Hello, everyone. I'm Christy Risk, Senior Editor at Genome-wide, and I'll be your moderator for today. The title of today's webinar is Genome-wide Study Reveals a Pseudogene-Associated Recurrent Gene Fusion in Prostate Cancer, and our sponsor is Advanced Cell Diagnostics. Our panelists today are Dr. Palanisamy, Associate Scientist and Professor in the Department of Urology at the Vattikuti Urology Institute in the Henry Ford Health System, and Dr. Anushka Dikshit, Application Scientist at ACD. You may type in a question at any during the webinar. You can do this to the Q&A box, which appears on the right side of the webinar screen. We will conduct the Q&A portion of the webinar after the presentations have concluded. And if you look to the tray at the bottom of your window, you'll see a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Dr. Palanisamy. Please go ahead.
Hello, everyone. I'm Nallasivam from the Henry Ford Health System. Today, I'll be speaking to you about our recent work on the discovery of new pseudogene-associated recurrent fusion in prostate cancer. And this work was published with the extensive next-generation sequencing analysis of a large number of tumor samples from different solid cancers, including prostate cancer. As you see here, using next-generation sequencing technology, one can do several type of kind of sequencing analysis, including whole genome sequencing or whole transcriptome sequencing, which we call RNA sequencing, and recently the most popular target capture and whole exome sequencing type of analysis. So given the enormous complexity of the cancer genome and the extensive copy number aberrations, when we do the whole genome analysis, there will be a lot of mutations and aberrations which are very difficult to comprehend, to associate with the tumor development. So therefore, in order to reduce the genomic complexity, we focused our analysis on the transcriptome sequencing, or other words, just the mRNA sequencing. So we did the whole transcriptome sequencing. So using the sequencing reads, one can perform several type of analysis. Since the next generation sequencing is a type of analysis is heavily unbiased, unlike the gene expression microarray, so that we will be able to identify several non-coding RNA or differential gene expression or a mutation discovery. We can identify some novel transcripts that are not annotated in any genome database, and then we can discover novel gene fusions, alternate splice variants, and sometimes you can look for some viral genome as well. So besides the well-known non-coding RNA and the protein-coding genes, our work was the first type of analysis. We looked at the expression of pseudogenes in prostate and other cancers on a genome-wide scale. Although several anecdotal evidences have been presented in the literature on the discovery of individual pseudogenes like the P10 and other pseudogenes, this is for the first time we conducted a whole genome-wide analysis for the expression of pseudogenes across several solid cancers, which I will present in detail in the subsequent slides. Before going to that, briefly I would like to review the expression of the transcriptome sequencing in our whole genome. If you look at the differential sequence components that is making up the human genome, out of the 3 billion base pairs that constitute the human genome, the protein coding genes constitute only about 1.5% of the entire genome. And the rest of the sequences, we thought they were junk until the human genome sequencing project was completed. Because those sequences, we don't attribute any functions, therefore those sequences were called junk. But with the whole genome sequencing project was completed, we were able to discover several types of long non-coding RNAs, which constitute a majority of the human genome. So now I think we actually changed our view that human genome is transcriptionally active, like more than 90% of the human genome is transcriptionally active. including both protein coding genes and non-coding genes. So even in this slide, when you see it, there is no mention about the pseudogenes because of the complexity in the analysis of the pseudogenes. I'll describe more in detail in the subsequent slide. No attention was paid looking at the pseudogenes. So even before the sequencing analysis of the human genome, we thought even the non-coding RNAs were also junk sequences, because since they are not coding for any particular protein, we thought that these non-coding RNAs do not have any functional consequences. But in the recent extensive analysis of several normal genome and several cancer genome, we found that many RNAs show abnormal expression pattern that are specific in cancer and different from the benign tissue, and they do play an important role in the regulation of many protein coding genes as well. So the change, all these analyses that changed our perception of non-coding RNA from being junk transcriptional products to functional regulatory molecules. So like that, recently with the advent of the next generation sequencing, there is a considerable interest in the research community looking at the pseudogenes. So the pseudogenes are again a class of non-coding RNA, but they are totally distinct from the long non-coding RNAs. The term pseudogene was first used only in 1977 by Jack et al. They identified this pseudogene in an African clawed frog named Genopus levis. It is the first description or evidence to show the expression of the pseudogenes. So in order to define the pseudogene, I would like to make the distinction from long non-coding RNA. So basically the pseudogenes are dysfunctional relatives of known genes that have lost their protein coding ability or not expressed at all. So essentially, these are all, the pseudogenes are nothing but a derived sequences from the already functioning gene. Okay, so the sequence similarity will be quite identical, but with a few mutations and other things, which I will describe more detail in the subsequent slides. So any genomic sequence that is similar to another gene and defective, meaning that they are not functional or not expression, is called a pseudogene. So there are three major types of pseudogenes based on their expression and the genomic organization. The major, about 72% of all the pseudogenes are classified into processed pseudogenes, meaning that a portion of the mRNA transcript of a gene spontaneously reverse transcribed back into DNA and inserted into different regions in the genome. So this is called processed genome. So essentially, after the initial transcription of any gene, the intronic sequences are removed. After the splice processing, only the exons are fused, and for some reasons we don't know the exact mechanism, in the cell itself, the RNA is spontaneously reverse transcribed and made into a strand, and that get inserted into a different in the genome. Although the exonic sequences are similar to the wild-type gene, but this new pseudogene that lost all the intron and exons, so that you will see what in other words, we call as intronless genes as well. So the next category is non-processed pseudogene. That means here there is no transcription, but a segment of the genome is duplicated. That again occurs since our human genome is highly dynamic structure. It goes several changes during the evolution. So a section of the human genome is duplicated and copied, and it gets inserted into a different location in the genome. So this is called non-processed. So essentially, these type of pseudogenes, they retain all the introns and exons, but they may not carry the regulatory elements, the 5' promoter and the other regulatory elements. Because of that, these non-processed pseudogenes are not expressed or dysfunctional. And the third category is the disabled pseudogenes. So these are all the genes neither transcribed or not duplicated, but these genes are located in the same location. But what happens was they acquire mutations at the regulatory regions. Because of that, they lost that transcriptional ability. So these are all called disabled pseudogenes. So these are the three major categories of pseudogenes that have been described in the literature. So here in this slide, I show you in the picture view of the three or one new category of pseudogenes. So as you see in the figure A, this is the example of the processed pseudogene where mRNA is transcribed into a single-stranded RNA with the poly A tails and everything. Then it gets spontaneously reverse transcribed into double-stranded DNA and get inserted into a different location. This is called processed pseudogene. And in figure B, we have the unprocessed pseudogene. So here again, it has all the introns and exons. But if you see the red vertical line shows the mutations that are acquired in the duplicated segment of the DNA inserted into different locations. Because of these mutations, they become different and they are not functional. So this is unprocessed pseudogene. And the third category is the unitary pseudogenes. That means they are located in the same location, but they acquire some new mutations during the evolution and they become non-functional. So this is the third category. And recently there is a fourth category that is actually described in the one of the recent reviews in the Nature Reviews Cancer, where they showed there is an obvious difference. So originally, when you look at the human genome reference sequence, you will see they are annotated as pseudogene with all the mutation, everything. But in the new sequence, what they see in the non-reference sequence, those mutations are lost. Okay, this is a new kind of category that is emerging. I think it's all because of the next generation sequencing data that is available both at the whole genome and whole transcriptome level. Then we go to the function. What is these pseudogenes are really doing? If they are not protein coding, if they acquire mutations, what exactly they are doing? So based on some of the anecdotal evidences, the pseudogenes do have a function, like either they function as a protein, because we thought that the pseudogenes are disjunct or junk DNA, but now we have enough evidence to show that the pseudogenes indeed they do transcribe into messenger RNA and they do make protein. So they function as a protein. So some of the pseudogenes indeed, they express protein, or some of the pseudogenes they acquire some mutations called a truncation mutation that will create early stop codon, so that might result in the production of a truncated protein. So there are examples for these as well. And RNA based, sometimes the transcribed the pseudogene may function as a RNA that will regulate the function. So the way it is functioning is, as I mentioned before, since the pseudogenes are derived from the already existing gene, because of the sequence similarity, when both the wild type gene and the pseudogenes are expressed, the pseudogene will compete with the wild type RNA, and they may kind of repress the expression or the translation of the protein. That's how the function is exhibited there. And the third category is the DNA based. So they, in terms of chromatin interactions, they may interact with the different locations in the human genome. They may regulate the transcription of a gene or the function of the gene. Based on the 3D chromatin interaction analysis, people have shown that it can function as a chromatin regulation as well. And the other way is some of the pseudogene segments they get actually as inserted into the wild type gene, altering the wild type gene as well. So these are some of the examples of the functions that are described in the literature based on anecdotal evidence. But still, the whole genome-wide expression of the gene and the function is not completely annotated, but a lot of work is, a lot of interest is shown recently, and I'm sure in the future there's more evidence will come up. In the next slide, I'll show you. I will discuss some of the key challenges in the identification of pseudogene. So as I mentioned before, since the sequence homology with the wild type gene and the pseudogene is high, separating the sequence reads from the pseudogene and the wild type gene is a little challenging. That is bioinformatically challenging, but there are some improved versions of the software are available now. So the delineating the unique signature from highly homologous sequence is a challenging part. It is because of the close similarity between pseudogenes and its wild type gene family have many homologous wild type genes. And I will show you some examples. It is not always like one pseudogene is derived from a wild type gene. Sometimes more than one pseudogene will be generated over a period of several million years of evolution. We found that some genes have more than like 15 or 20 pseudogenes located in different locations in the genome. And the other challenge is the processed pseudogenes are often identified by the lack of introns, but some protein coding genes also lack introns, what we call as intronless genes. So already well characterized or well annotated genes are also found to be intronless genes. Since the processed pseudogenes do not carry the introns, so sometimes it is often challenging to separate whether to call it as a pseudogene or as a protein coding gene. So this is again another challenging. Some pseudogenes, they lack stop mutation that truncate the protein and thus have the same protein coding capacity as the parental gene. So because these mutations that are acquired in the pseudogenes, it may not affect the open reading frame. So if the pseudogene is expressed and it is making protein, it might be similar to the wild type gene with, of course, with a few amino acid changes. It could be synonymous or non-synonymous mutations depending on the case that we are looking at. So these are some of the challenges associated with actually analyzing pseudogenes. So the main requirement for better analysis is we need to have really high resolution, in-depth sequencing, minimum 100x or more than that, in-depth sequencing. And essentially, the sequencing for all the samples should be generated from a single sequencing platform. So that is these are the key for successful analysis of pseudogenes. And it is preferable to look at the transcriptome sequencing or RNA-seq rather than looking at the whole genomic DNA. Because the genomic DNA, it is very difficult to separate the pseudogene from the wild-type gene. It is always looking at the transcriptome RNA sequencing where you can actually see the expressive pseudogenes that you can actually separate. I will show you some examples in the next slides. So I think enough for the introduction. So I will show what we have done in our analysis. So this is for the first time. That is in 2002, we published this work in Cell, where we built a compendium of about 248 cancer samples with matched benign samples across 13 different solid cancers, with including large number of cases for prostate, and we did an extensive analysis for the expression of pseudogenes. Here, even in this paper, we described the pseudogene pipeline, how we actually separate the pseudogene reads from the wild-type gene sequence reads. So here essentially you need the paired end sequencing with the long reads. So all the mate pairs are, when you have these mate pairs, you map to the reference genome. So when the mate pairs are mapping to the already annotated genes, you can separate all those reads, but select only the reads that are actually like one mate pair is mapping to a known gene and the other mate pair not mapping to any unmapped region, or sometimes sometimes both mate pairs may not map to any known locations in the human genome because the ends of these mate pairs are different, so that you may not be able to map these reads into the reference genome. So these are all the type of reads that is needed for pseudogene analysis. So we have to be careful that we have to remove all singletons. So we have enough reads for each gene, and make sure to remove all the stacked ends, because the ends of the sequence should not be the same between reads. So those type of reads have to be eliminated. So by filtering all those noises, we will be able to select the sequence reads, and that can be queried against some of the pseudogene databases that are established in the Yale University and other consortium. There is a database separately available for pseudogenes, and it can be mapped against those databases instead of the normal reference, human reference sequence. you'll be able to identify those pseudogenes. So in the next slide, I will show an example how we separate it out. Using this pipeline, I'm showing an example of ATP8A2. This is a pseudogene that is commonly expressed in breast cancer. So in the top line, the wild type, that is the ATP8A2 gene, and the pseudogene sequences is actually listed below. As you can see, all the red, all of the sequence reads in the red are the mutations that are acquired in the pseudogene. So as you can see, the sequence is highly kind of homologous with the wild type gene. Similarly, another example in the prostate cancer, we found a CXADR, that is a wild type gene, and we found the pseudogene, and there is a lot of insertion and deletion. So some of the sequences are missing in the pseudogene. So these are all some of the fundamental differences between the normal sequence and the pseudogene sequence. That analysis of these 13 different solid cancers, we were able to identify about 2,000 pseudogenes to be transcribed either ubiquitously or in a tissue cancer specific manner. Okay, so we were able to filter and we were able to cluster based on the manual curation and in-depth analysis. We were able to identify pseudogene expression that are expressed only in a particular tissue or only in benign or in cancer. So like that, in the last, this on the rightmost figure, we show the separation of of pseudogenes that are expressed in a cancer-specific manner, and the genes listed in the red box are the pseudogenes that are expressed in prostate cancer, that I will show in the next slide. These are the 8 candidate pseudogenes that we identified to be expressed in prostate cancer. The RAN, MTCH1, CXADR, and other genes. As you can see, some of these, like the RAN gene, has 15 pseudogenes, and they are located in chromosome 7, 8, 11, like that. Okay, so anyone, if you have the UCSC browser, you can actually visualize locations of genes, either processed or unprocessed, like that. So what we found was, in our pseudogene genome-wide analysis, even though a single gene may have more than one pseudogene, it's not necessary that all the pseudogenes are expressed. So we found that, for example, CXADR in prostate, out of these 3 pseudogenes located in chromosome 15, 18, 21, we found only the pseudogene on chromosome 15 is expressed and the other 2 are not expressed at all. So it seems like there is some selection, and it is like any other genes, they all, that there is some selection and they have some functional consequences why they are expressed in particular cancer, like that. So this will give you an idea, what you are looking for when you are doing pseudogene analysis. You're not dealing with just one pseudogene, so you may see multiple pseudogenes for a single wild type gene. So besides identifying these 8 candidate pseudogenes that are expressed in full length, what we found was, interestingly, we found since gene fusions are associated with several cancers, like the BCR-ABL gene fusion in chronic myeloid leukemia, TML4-ALK in lung cancer, and TMPRSS2 in prostate cancer, what we found was, interestingly, we found a new gene fusion that involves one protein coding gene and a pseudogene, and that is expressed specifically in prostate cancer. So that I will talk more in detail in the subsequent slides. So here I show you the gene fusion that we identify involves a KLK4, which is a protein coding gene. Then the KLKP1 is the pseudogene that is derived within the kallikrein gene cluster on chromosome 19. Okay, this within the red box I show the location of KLK4 and KLK2. So between KLK4 and KLK2, the KLKP1 is located. And the first, I will show in the next slide, the genomic structure fusion. The KLK4, it contributes the first 2 exons of KLK4 actually fused with the exon 4 and 5 of the KLKP1. The KLKP1, the full length KLKP1 contains 5 exons. It is not coding for any protein by itself. The normal full length KLKP1 not coding for any protein. So now what happens was the first 2 exons of the KLK4 fused with the exon 4 and 5 of the KLKP1 that resulted in the fusion, and we found that based on the open reading frame analysis, we found that indeed this fusion transcript is coding for a protein. So we identified 165 protein. So what is interesting is this is the first time, this is a rare and a new phenomenon that we observed in any solid cancer, and the first time in prostate cancer. What we found was the non-coding pseudogene after the fusion is converted into a protein-coding gene. So here the amino acids that are shown in blue are coming from the protein-coding gene KLK4, and the red amino acids are in fact coming from the pseudogene KLKP1. So this is the first evidence where we show that because of the fusion, the non-coding pseudogene is converted into a protein-coding gene. So we are very intrigued by this finding. So then we designed a primers based on the fusion junction, and we screened several prostate and non-prostate samples, and we found that in the figure A, the histogram shows the expression of the fusion gene only in prostate cancer, but not any other solid cancers like the melanoma or the breast cancer, and even it is not expressed in benign. It is very low level. It could be due to the contaminating kind of tumor tissues within the so-called benign tissues, but still the expression is not very high, but we see very high level expression only in prostate cancer, but not in any other cancer. This is an interesting finding. So based on this, we wanted to actually characterize the function and other aspects of this fusion gene. So going to that, what we did was, as I mentioned before, the KLKP1 full length is not making into protein. So we looked at the expression of this KLKP1 full length in the GTEX portal. As many of you are aware, the GTEX portal contains a next generation sequencing data of more than 50 normal human tissues. You can look at the expression. If you are interested in any particular gene in cancer, and you may want to look at the expression of this gene in a benign tissue. So with this type of analysis, in this slide I show the full-length KLKP1 is expressed only in prostate tissue, not in any other 52 tissues. So which shows the specificity or the importance of this KLKP1 in prostate tissue. But what we found was the fusion, the KLKP1 after fusion with KLK4 is expressed only in prostate cancer tissue, but you don't find the fusion in the benign prostate, as I showed you in the previous slide. So that adds more functional importance for the role of in prostate cancer. So here I will show you in this slide showing the genomic organization of this fusion gene. The top figure A, it shows the first two exons is fused with exon 4 and 5 of the actual sequence read that we actually mapped back to the UCSC genome browser. This is the location. And the figure B is the full-length cDNA sequence. The red sequence and the blue clearly shows the non-coding region of the gene. And the ATG to the stop codon is the coding region that is translated into protein, which is about 165 amino acids. The amino acids in black are from the KLK4, and the amino acids shown in the red are coming from the pseudogene, so which constitute this fusion protein between KLK4 and KLKP1. So next we wanted to actually characterize the function of the pseudogene. Okay, so we want to validate the expression of the pseudogene in prostate cancer. So since this is a pseudogene, there is no good antibody or anything is available. What we did was we used the RNA scope or the RNA in situ hybridization that is developed by the Advanced Cell Diagnostics or the ACD Bio. So I collaborated with them, and they helped me to design the antisense oligo probe that is specific to this fusion transcript. Using this antisense oligo probe, we were able to screen a large number of prostate tissue. So how the RNA scope technology is working, the technical aspects will be actually described by Anushka Dikshit in the next presentation. After my presentation, she will explain how the probes are designed, how the specificity is ensured, and she will explain more details. So using this RNA scope probe, we were able to screen a large, more than about 650 samples that are on a tissue microarray, and we clearly show that the expression is only in the prostate cancer tissue, as I show in the D on figure 1. there is no expression on benign tissue on the left two panels, and the right two panels are the cancer tissue where you see expression. At the bottom, in C, we show that the differential expression or different level of expression. Some cancers show the low level expression, as we show in the PCR data in the 2 slides before. So some samples show very high level expression, which we classify into like a 1+, 2+, 3+, or 4+, like that. So based on this expression, we were able to show that the expression is very specific to high Gleason grade prostate cancer that we show in E, and and in the F we show that this gene fusion is expressed particularly in men who are under the age 50. Young men with prostate cancer are shown to have more of this gene fusion, and we found that since prostate cancer has several other gene fusions like the TMPRSS to ERG, we found that the KLK4 fusion is expressed more in the ERG fusion positive cancer. There's more like concomitant expression. And also we looked at the racial disparity perspective, whether both African American and Caucasian show, and we found that there is an equal level of expression in both race, but we found more patients from the Caucasian kind of prostate cancer show more number of cases, but the difference is not statistically significant. So the next slide is to show the concomitant expression of KLK4 fusion in the ERG positive tumors. On the right side, we did the immunohistochemistry using the ERG antibody on the expression of this, all the green signal shows the ERG protein expression only in the tumor tissue but not in the benign. Similarly, using the RNA scope, we showed that the same locations wherever the cancer, we see the expression of KLK4 fusion as well. So this is, since we can do this RNase, we don't have to do it under fluorescence mode. It can be done under bright field, so it will be easy to interpret the results like reading the immunohistochemistry. So that is the power of this RNA scope, which anyone can take advantage of this. So the next slide shows that we can screen a large number of cases using tissue microarray. Only the cases that are positive for the expression are showing the expression, but when the cases are negative, you don't see any signal at all. So that shows the sensitivity and specificity of this probe. Next, what we did was, since the amino acid sequences are coming from the pseudogene part, we decided to, since there is no antibody available, we decided to develop an antibody using the antigenic peptide, the CTI-SA, the antigenic peptide. We were able to develop a polyclonal antibody that is specific to this fusion protein. Using that antibody, we validated to confirm whether this fusion gene is indeed making a protein in the cells and tissues. So for that, we made a tissue kind of gene expression construct in the expression vector. The entire fusion gene is cloned into a vector, and they are transfected into RWP cells. In the figure A, it shows the overexpression of the fusion transcript. Then the Western blot, because this fusion transcript, when we made the construct, we put the N-terminal flag tag sequences so that we can use the anti-flag antibody to confirm the expression of the protein. So in fact, in figure B on the top panel, which shows the anti-flag antibody recognize the fusion protein. Then again, using our fusion-specific polyclonal antibody, also recognizing the same band. And in the figure E, we show the full length or the full blot of the Western blot, which shows the specificity of the signal there. And also on the immunohistochemistry, we show that the tumors that are confirmed to be the fusion positive, confirmed by PCR, is positive by IHC, but the tumors that are negative did not show any staining. So that shows the specificity of both antibody as well. And in the next slide, I will show you with functional characterization. Since we know that this fusion transcript is making protein, and we wanted to show whether this fusion gene is oncogenic, so we did invasion and proliferation assays using the RWP and PREC cells, and we show that overexpression of this fusion gene in RWP and PREC cell shows increased proliferation and invasion. And we also did the chicken chorioallantoic membrane assay to show that whether and the tumor has any potential for metastasis, which clearly show that there is an intravasation to lower CAM in the chicoria electronic membrane as well. And we looked at the gene expression analysis of the RNA isolated from the cells transfected with this fusion. We found the differential expression of the genes associated with the cancer metastasis, which shows the importance of this fusion gene in developing prostate cancer metastasis. And we also screened a large panel of metastatic prostate samples that are converted into patient-derived xenografts. So we screened about 31 patient-derived xenografts with collaboration with the Dr. Narayan from MD Anderson Cancer Center, and we were able to show the endogenous expression of this fusion gene in the patient-derived xenografts, which are a good resource for doing additional functional studies. Then since this is a prostate cancer specific pseudogene, we asked the question whether this fusion gene is regulated by androgen, like several other fusion genes. And in fact, we found that there is a strong androgen receptor binding site and the ERK binding site at the fusion junction before the exon 4 of this KLKP1, and there is another strong ERK binding site in KLK4 as well. So this confirms that indeed this fusion gene is regulated by androgen. And also in this slide, we looked at the location of the protein, the fusion protein, by using the fluorescent anti-flag antibody. We showed that the nuclear localization of this fusion protein, like we see the RNA signal on the left panel, the localization of the signal in the nucleus. Then, like other prostate cancer gene fusions, we wanted to see whether this gene fusion can be detected in the urine of the prostate cancer patients. Since this fusion gene is prostate cancer specific, not in benign, we want to develop as a non-invasive diagnostic assay based on the urine samples, and we screened random samples, and we were able to show that indeed we can see this gene fusion in the urine of the prostate cancer patient. So our goal is to develop this into a non-invasive kind of cancer detection assay. So with this, we will be able to detect prostate cancer even before taking the biopsy. So this is inconsistent with several other kind of prostate markers, including the TMPRSS2-ERG or the PCA3. There is already an FDA-approved test is available for PCA3, but combining with the TMPRSS2-ERG and KLK4, one can reliably or categorically, you can say that you can detect prostate cancer. Since the prostate-specific antigen or the PSA test is a controversial one, because elevated PSA doesn't mean that the patient indeed have cancer. So I think these markers can be combined with the elevated PSA to make a definite conclusion that whether the patient has a cancer or not. So that is our main goal. We are working towards that goal. And all results are published in this paper. We published in October 2019 in Neoplasia. So you will find more details about the gene fusion in this paper. So with that, I will conclude my presentation. I hope I have shown enough data how you can actually discover. We showed the pipeline for the discovery of the pseudogene and how we can make an important discovery by doing this kind of analysis by looking at the uncharted territory of the pseudogene. So this is our first kind of data showing the genome-wide analysis of pseudogene expression, and we found an important marker, cancer-associated marker, which has a potential for kind of diagnosis. So all these people who actually really helped me in this project, I would like to thank all of them and the funding agency. So with that, I will conclude my presentations. I will actually hand over this presentation to Anushka Dikshit, who is from the patient scientist from Advanced Cell Diagnostics, and she will explain more in detail about the RNase technology. Thank you for listening. I will stay with this presentation after Anushka. I'll be happy to answer any questions. Thank you.
Thank you, Dr. Palanisamy. As a reminder to webinar participants, if you have a question, please type it into the Q&A box. We will conduct the Q&A portion of the webinar after the presentations have concluded. Next up is Dr. Dikshit. Please go ahead.
Thank you for that introduction. After that brilliant presentation by Dr. Palanisamy, I will now talk about the applications of RNA-scope technology, specifically in cancer research. For those of you who are not familiar with the RNA-scope technology, it is an in situ hybridization technique that was specifically designed to overcome some of the traditional limitations of an ISH assay while still giving you high sensitivity and specificity of detection. The three major parts of this assay are the target probe design, where we design specific target probe against the gene of your interest, the signal amplification strategy, where we have developed a strategy that gives you really high signal-to-noise ratio, and then detection and quantification, where we allow detection of a single molecule of RNA, either in a chromogenic or fluorescent platform. This technology can be applied to a wide variety of sample types, including FFPE tissues, fresh, fixed, frozen tissues, cultured cells, PBMCs, and so on. Additionally, our technology can be applied to different target types like mRNAs, long non-coding RNAs, splice variants, CDR3 sequences on T cells, point mutations, and so on. In this method, I will focus a bit more about the probe design and what makes this technology highly sensitive and specific. As you can see on the slide, we depict this RNA-scope probe as a Z, mainly to highlight the three different parts of this probe: the preamplifier binding site, the linker region, and the target binding site. As the name suggests, the target binding site is where the probe binds on to your transcript of interest, and the preamplifier binding site is where the preamplifier binds and forms the signal amplification tree. Now, the two Z probes have to bind in tandem to one another on the target sequence for the preamplifier to bind on to the probes, after which the amplifier and the labeled probes bind on to this to form your amplification tree. This gives you high sensitivity of detection, which allows detection of really low expressing targets. Now, how do we ensure specificity? If the two Z probes do not bind in tandem onto your target, then the preamplifier molecule cannot bind onto the probe, and there is no tree formed. As a result, there is no signal detected. So this ensures high sensitivity and specificity, giving you high signal-to-noise ratio. In this particular slide, we've depicted all the different types of assays that we offer based on the target of interest that you want to detect. We offer our assays in chromogenic and fluorescent platforms. You can detect 1, 2, 3, 4, or now up to 12 targets using this technology. This technology is also available as a manual form of assay, or it can be done on an automated system like Leica or Ventana. Currently, we have over 25,000 probes in our catalog, but technically, we can design a probe against any species and any target as long as we know the sequence against which we want to design the probe. The two major assays that we offer are the RNA-scope assays and the PACE-scope assays. The RNA-scope assay is mainly for targets that are over 300 nucleotides in length, and the probes that we design are normally a pooled probe of 20 ZZ pairs. The PACE-scope assay is a more specialized assay that was designed to detect targets like splice variants, highly homologous sequences, and point mutations. And the probes that we give here are normally smaller in size and up to 1 to 3 ZZ pairs in length. RNA-scope has been applied to a wide variety of research areas, including basic science research, infectious disease research, cancer and clinical research, as well as biopharma discovery and development. As you can see on the screen, we can use this technology to detect a wide variety of targets, like non-coding RNA, splice variants, GPCRs, point mutations, and so on. For the purpose of today's presentation, I'll be focusing on the applications of RNA-scope in tumor biology and immuno-oncology. Additionally, using our pharma assay services, we can do the assays for you, do the data analysis, and provide the results back to you within 4 weeks of receiving the sample. Now, focus on the different applications of RNA-scope in cancer research. Starting with application of RNA-scope in characterizing immune cells and soluble factors. In this particular example, one of our customers used RNA-scope to detect the expression of chemokine CXCL9 as a marker for activated immune cells. They compared two different lung cancer cell lines and demonstrated that the cell line with active interferon gamma signaling had a better response to immunotherapy. When they potentiated interferon gamma signaling in the resistant cell line, they observed increased responsiveness to anti-PD-1 therapy and increased immune cell activation indicated by CXCL9. In addition to detecting soluble factors, we can combine our RNA-scope-ish technology with IHC or IF, which allows visualization and detection of RNA and proteins simultaneously. This can be extremely significant if you're trying to characterize complex tissue structures, identify origin of secreted proteins, and so forth. Here is a good example of how we have visualized the expression of different markers to characterize immune cells in cervical cancer tissue. Using the CD3 antibody, we have identified the T cells, and then using RNA-scope-ish, we have visualized gene expression for interferon gamma, PD-1, and PDL1 within these immune cells. In addition to doing this on a different platform, we can also do this dual-ish IHC technique using a chromogenic platform. As you can see in this particular example, where we have visualized the expression of CD68 using the CD68 antibody in this first panel, potentially detecting macrophages, and then looked at the expression of IL-12 and CXCL9 using RNA-scope. In the second panel, we have identified CD4 positive T cells using the CD4 antibody, and then visualized TGF-beta and FOXP3 using RNA-scope-ish. Moving on to our second application, we will now look at the application of RNA-scope in visualizing tumor heterogeneity. In this particular example, we are visualizing PDL1 in lung cancer tumors, and as you can see, all these the four different samples that you can see express different degree of PDL1 expression, highlighting the inter-tumor heterogeneity of this particular marker, which can be nicely visualized using RNA-scope red assay as shown in this image. In addition to the inter-tumor heterogeneity, we can also visualize the intra-tumor heterogeneity. As you can see in this image, PDL1 especially is extremely heterogeneous. As you can see in the same tumor, the degree of expression of this marker varies dramatically based on the region that you're visualizing. And this can be extremely significant if you're trying to develop immunotherapies against specific targets, in this case, PDL1. So this technique allows you detection at a single cell and single molecule level and allows you to understand the heterogeneous expression of specific markers. Next, we are going to look at the application of RNA-scope in detecting splice variants. Now, splice variants are extremely significant when it comes to cancer research because they can be drivers or initiators of certain cancers. In this particular example, this group of researchers wanted to visualize the expression of delta-16 HER2 splice variant of the HER2 receptor. And this is significant because individuals possessing the delta-16 HER2 splice variant are extremely sensitive to Herceptin therapy. So they used this strategy to distinguish between patients carrying the splice variant versus patients that had the wild-type HER2 receptor. And the probe design strategy that they used is they designed a probe that either spanned the exon 15-17 junction to detect the delta-16 HER2 variant, or designed a probe that spanned the 15-16 or 17-18 junction, which will now detect the wild-type HER2. So based on the type of probe you use, you can identify if the patient has the splice variant or the wild type. And they were able to distinguish these patients based on their presence of the splice variant and stratify them for Herceptin therapy. Similar in this particular case, this group of researchers used the BaseScope assay to visualize the expression of circRNA of androgen receptor. Now, this particular splice variant of androgen receptor is seen to be highly expressed in this castration-resistant prostate cancer compared to a less aggressive prostate cancer. Again, highlighting the role of this particular splice variant in promoting growth of this prostate cancer tumor. We look at the application of the RNA-scope technology in validating single-cell RNA-seq data, which has come about as a significant technique last decade because it allows for detection of RNA expression at the single-cell level and gives you high throughput data analysis. In this particular example, this group of researchers from Harvard wanted to analyze glioblastoma tumors, and when they performed single-cell RNA-seq analysis, they identified 4 different cellular states in which glioblastoma tumor cells can exist. They identified specific markers associated with each of the states. And using RNA-scope, they validated the presence of these cellular states by using probes against the specific markers. But what they identified instead was that the glioblastoma tumor cells can not only exist as a single cellular state, but they can exist as a hybrid state, as you can see in this RNA-scope staining panel on the right side. They used the duplex assay, and what they identified was they could visualize the hybrid states of these glioblastoma cells using specific markers, which was an interesting finding and will aid in developing newer and better therapies for treatment of this deadly cancer. Let's talk about the significance of RNA-scope in identifying specific T-cell clones, which is very significant since we are developing more and better engineered T-cell therapies for cancer. In this particular example, we use the BaseScope assay to detect specific CDR3 regions on specific T cells. Here we've compared the CCRF cell line with the Dikshit cell line, and we designed probe specifically either to the Dikshit CDR3 or specifically to the CCRF CDR3. And based on which probes we are using, we can see that positive signal can only be visualized when we use the Dikshit probe in the Dikshit pellet or when we use the CCRF probe in the CCRF pellet, just highlighting the specificity of our BaseScope probes in identifying the CDR3 regions. And a great example of how this was utilized was here. In this particular example, this is a group from Vanderbilt that was trying to understand the reason for anti-PD-1 therapy-induced encephalitis in this particular patient that had melanoma. And this patient succumbed to their encephalitis. What they wanted to identify was the cell population, immune cell population, that was responsible for inducing this encephalitis. And when they performed TCR sequencing on this encephalitis tissue, they identified a specific sequence that was most abundant in this particular tissue. Then they went ahead and designed a BaseScope probe against this sequence. And what they found was that this particular TCR was co-localized with CD4 cells and not with CD8 cells, highlighting that this population that was responsible for inducing encephalitis was a CD4 positive T cell population, which they later went on to identify as a memory CD4 positive T cell population. And BaseScope played a very significant role because it allowed visualization and co-localization of the specific TCR with CD4 cells, and mainly showed that this was not co-localizing with CD8 cells and built confidence in understanding the cell population responsible for inducing this encephalitis. Finally, here we show how well adopted our technology is and how we've grown in the last few years. As of today, we have over 2,600 publications in different research areas. Cancer and neuroscience are our top research areas with over 800 plus publications in each of these research areas. Important fact, over 25% of our papers are published in 3 big journal publishing houses of Nature, Cell, and Science. Just again, it shows you the quality of our publications and how this technology might help advance your research especially in cancer and neuroscience. Finally, if you have any more questions, do not hesitate to reach out to us on the email below. Thank you.
Thank you, Dr. Dikshit. A reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to give us feedback by taking our exit survey after the webinar has ended. All right, our first question, Dr. Palanisamy, are you proposing that mutations occurring in the wild type of the gene and then their conversion into the pseudogenes could be the culprit in the genesis and progression of cancer?
Yes, as I showed you very clearly, the KLKP1, the full-length pseudogene, is not expressed in prostate cancer tissue. It is specifically expressed only in the normal prostate. Like any other gene fusion that have been described in different solid cancers, this KLK4, KLKP1 gene fusion identifies a new molecular subset of prostate cancer. And as we showed, the oncogenic property or the experiments that clearly show that this gene fusion indeed has the potential to transform the normal cell into a cancer cell. Yes, to answer the question, yes, indeed, this gene fusion has some functional role in the prostate cancer development.
And for the detection of pseudogene fusions, in your experience, does the next generation sequencing software pipeline need to be trained to look for the pseudogenes and then to call them?
Yes, I think the next generation sequencing technology, as I showed you in the first slide, it offers several advantages. One can do several different type of analysis as long as they have high resolution, in-depth, and data generated from a single platform. One can do all different type of analysis, particularly for pseudogene. And that will be a little challenging based on some of the reasons that I have actually described in my presentation. It will be challenging. There is no kind of automated pipeline have been developed so far. As I showed you in the figure, one has to do a lot of manual curation analysis by systematically filtering the reads to separate the pseudogene reads from the rest of the reads. And yes, it is informatically challenging, but it is not completely, not doable or anything. One can actually be patient, and if they have interest, they will be able to identify these cancer-specific genes. But right now, there is no kind of open source kind of data analysis pipeline for pseudogene has been developed, but one can easily follow based on the methods that we have described. And I think you will find more information when they looked at the paper published in Cell in 2012. Next question.
And for patients who have castrate-resistant metastatic prostate cancer where the solid tumor has been removed, what would you say the implications of this discovery are for developing curative therapies, for example, through immunotherapy?
Yes, as we have clearly shown that we screened several metastatic castration-resistant prostate cancer that are converted into patient-derived xenografts, we clearly show that more than about 20 or 25% of this fusion is expressed in metastatic prostate cancer. Even after the patients undergone radical prostatectomy, that means after removing the prostate, it is possible there are actually a significant number of cases they come back with the metastatic disease. So which means that even before removing the prostate by radical prostatectomy, it is possible that the cancer cells they established or they escaped from the main prostate gland and established metastasis. Sometimes the patients who have a recurrent metastatic disease, initially when we screen the primary tumor, if they are actually positive for this gene fusion, and this gene fusion can be used as a marker to screen the recurrent metastatic disease. And if the patients are undergoing treatment, yes, using this marker, we can actually monitor the response to the treatment. If at all any treatment is available for the metastatic prostate cancer, we can use this marker to see whether the tumor cells are going away. Yes, this is a very important, like any other marker, like EML4, ALK that people are using in lung cancer, and it has the equally importance that the gene fusion can be used to monitor the response to the treatment and also the progression of the disease. Yes.
Great, thank you. I'm afraid that's all the time we have for today. We'd like to thank Nallasivam, Anushka, Dikshit, and our sponsor, Advanced Cell Diagnostics. If we didn't have time to get to your question, we will follow up with our experts. As a reminder to webinar participants, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.